Thank you, Dan. Thanks to everyone for coming out on a cold winter's night. I have to talk here, sorry. I'm going to tell you about the results of somewhere in the neighborhood of 30 years of research on this wonderful group of animals, the Dasyuromorphian marsupials of Australia. It's been a wonderful career working on these things. They're inspirational organisms. They're charismatic in the most charismatic of all possible ways. And they live in the most exciting place on Earth, I think. It's been my good fortune to go chasing them around in their natural habitat. What I'd like to talk to you tonight about is one of the great insights that Darwin gave us. This is Darwin Week. It's Darwin's birthday. You remember that one of the great theses in The Origin of Species, one of the two great theses, was that organisms are related to one another by descent with modification. That existing species are derived by a process of ancestry. They're related through ancestry to pre-existing species. And one of the challenges, the great challenges to all of biology that Darwin laid out for us was then, if that's the case, to be able to reconstruct that ancestry, to infer it from the fragments of information that we have with us today, to basically try and go back through Earth history, through the history of life on Earth, and figure out who's related to who. I'm going to tell you about our efforts to do that with these 72 species, approximately 72 species, in this one order of marsupials. Let me maybe jump to the end. When you've been around this long, you sort of are indulged in the ability to give talks like this with a retrospective flavor to them. And although I am going to end by telling you about some of the most recent research that's been going on in our group, I'd like to go back and bring you up to speed, if you will. First of all, just by telling you something about the amazing biodiversity of these animals. This is an order, a big group that's all descended from a single common ancestor. And right now, taxonomically, we recognize three families in this order, three big branches of the Dasyuromorphian tree of life. And here's one of them. This is, to begin with, sort of a sad story. The family Phylogeny is no more. It is extinct. The last one you're looking at right there died in the Hobart Zoo in the 1930s. This family had a long and proud history in Australasia. At one point, there were as many as 5 different genera running around the rainforests of Australia back in the Oligocene, early Miocene. That diversity dwindled to just one species that lived in Tasmania when Europeans arrived, and within a few centuries, this animal was hunted to extinction on purpose to save that other amazing chunk of biodiversity in the world known as sheep. This is my favorite mammal. I have been fortunate enough to spend quite a bit of time with these little guys running around my toes. This is a numbat, the single species in the family Myrmecobioidae, with no fossil record, really no clue as to where this thing came from. This is the marsupial anteater. You may have heard of it as a spectacular example of convergence with placental mammals. We like to think of the placental mammals being the ones that converged on the marsupials. That seems more fair to me. They're restricted to southwestern Australia. And then there's the king of the hill in terms of modern diversity. These are the dasyurids, the 70-some species of marsupial carnivores and insectivores that currently dominate the top of the food chain in Australasia. So I'd like to explore a little bit of the family diversity within this big group, the dasyurids. I'll break it. It's 4 groups. It's 2 groups it's 2 groups each of 2 groups, starting with the name-bearing subfamily, the Dasyurinae, with about half the species in this tribe, the Dasyurinae. Again, these groups are all descended from single common ancestors. So the 26 species of Dasyurinae we know, and I'll show you evidence for this in a second, we know are each other's closest relatives. That's a mulgara, or two mulgaras. They're about the size of your fist. And I think what they're eating there is a cockroach. You see the background. These animals live in dry outback habitats in Australia, in arid environments. The Phascogalini, a much smaller group, but even more interesting in their own way. This is the group that you may have heard. This is the group of mammals where all the males die in a population after mating. This is the male die-off, or the male semal parity group. Fascinating lessons in evolutionary ecology from animals like this arboreal phascogale and their relatives, the antichinuses. The other big subfamily, the Sminthopsinae, with a relatively large number of species in this group, the Sminthopsinae, most of them are called dunnerts. Their common name is dunnart. That's a fat-tailed dunnart. This is a fairly cosmopolitan group across Australia, occurring in both wet and dry habitats. And I'll have more to say about the classification of this group in a few minutes. And some of the tiniest mammals on Earth, the planigales, those 5 species in a single genus, that creature right there is about the size of your thumb. About the size of your thumb, and that's the biggest one, the common planigale. Most of the other 4 are even smaller. These have the charming habit of hiding in the cracked clay soils in dry outback habitats in Australia. If you go looking for them, you look in the cracks in the clay. That's where you find them. So, what can I tell you about that's been going on recently? Well, this is an exercise in modern molecular systematics. Back in the good old days, may they never be forgotten, we used to do this as follows. We used to grab a few species, whatever we could get our hands on, usually tissue samples that were in somebody's freezer for some other reason. We would get a little bit of sequence from a few species and draw big conclusions from that information. And now, with the advance of technology, both biotechnology and analytical technology, we tend to get a lot of information, analyze it in incredibly sophisticated ways that sometimes we don't even understand, and come to a conclusion that is not that much different than what we knew already. And so that's kind of the story that I'm going to present to you here. But the good news is that we know a lot. Because of our efforts, we have quite a lot of information that is seemingly quite reliable about this group. These are my collaborators. The gentleman on the right is Mike Westerman, who is just a general force for good in the world. He is a driving force behind, or was a driving force behind, mammalian molecular systematics in Australia. Mike never passed a roadkill that he didn't stop and take tissue from. Just an amazing collection of material. And Mark Springer from UC Riverside, I think, has done more for mammalian systematics than any other person in the history of the world. And someday that will be widely recognized. The trouble is, he's made a lot of people mad. This is what we've done. We have taken basically all the species of dasyurids that there are, except 1 or 2 recently recognized ones that are perhaps dubious in terms of their validity, representing, the entire family, and both of the other families, the phyllocinids and the numbats. We've got a lot of information from 2 categories of sources. One is the mitochondrial genome, the little powerhouse of the cell, our ancient symbiont that carries its own DNA. We've got about half of the mitochondrial genome from these animals in terms of DNA sequences that we've obtained. There's the list of genes. And a roughly equal amount of information from the nuclear genome, from the chromosomes of these animals. That's a trivial fraction of what's in the nuclear genome, but it's still, by modern standards, a decent amount of information. There was a time when figuring out how to analyze data like this and produce a reliable estimate of relationships was something that got even philosophers excited. It taught us back about 20 years ago quite a lot about epistemology in evolutionary biology, and I'd love to go through that with you. However, right now, I'm going to sort of brush it aside and tell you that This is fairly sophisticated statistical analysis that relies on our ability to understand how the DNA sequences evolve on their own, and then use that information to make a reliable estimate of species relationships. And we do that using a couple of tried and true methods of estimation from statistics. The maximum likelihood method that has been around pretty much since the origin of statistics in the early 20th century. And then something that's even older, but only recently gaining in popularity, Bayesian inference. And yeah, if you know these things, great. If you don't worry about it. So, our most recent tree. Can you read those names? You don't have to. Let me just give you some highlights of this phylogeny. Here's the mouse. It starts down here. These guys out here and this little fella down. These are not Desiuromorphs. These are a bunch of bandicoots up here. And just things to help us figure out where the root of this tree is for the Desiuromorphs. But here's the ancestral Desiuromorph down here, and you see the earliest split on the tree gave rise to the phyllocynid ancestor, and then the ancestor of the other two families down here. After that, we see the split that gives rise to the numbat in purple, and then the big, big branch leading to Desiurids, all these Desiurids here. Within that group, there are the two subfamilies, the Desiurinae, the Spinthopsinae. Within each of the two subfamilies, the two tribes that I mentioned in each, the Planogalinae, the Spinthopsinae, and up here, the Phascogalinae and the Desiurinae. This was all very reassuring, because most of what I just told you, we got with the first 300 bases of one gene back in 1990. And now we're pretty confident that this is the correct pattern, with one exception that I'm going to tell you about. So you can see the color coding. All the oranges go together. Showing species derived from a single ancestral Phascogalin. All the reds go together. All these derive from a single ancestral Desiurin. All of the blues and all of the greens. And you notice there's some differential shading of the greens down here, which is something I'll tell you about in a second. So that's all really reassuring. If you're a mammal taxonomist, you could go through that with a fine-tooth comb, and you could say, boy, we're in good shape here, understanding the evolution of Desiuromorphs. But there are some lingering questions. As in any ongoing scientific investigation, these questions keep us up at night. Here's one. We have this big family, Desiurinae. We have these two other families, one extinct and one with just one species in it. What is the closest relative of the Desiurids? For a long time, we thought we knew. And the tree that I just showed you told us something completely different. The tree that I just showed you suggested that the numbats and the Desiurids are sister groups, as we call them, derived from a single common ancestor that arose after the separation of phyllocinids. That was not the answer we were expecting, because in this case, we thought that, for once, the anatomy of the animals was not misleading. If you look at these skulls, this is a thylacine here. You may have seen this skull in textbooks. This is a classic example of convergence of a dog-like carnivore set of adaptations. Those are big sectorial molars. The marsupial version, they have styler shelves along the outside. They have all the marsupial cusps, none of the derived placental cusps. Here's a quoll. That's like the first animal I showed you in the opening slide. And if you were to compare these two skulls point for point, you'd find a lot of similarities, a lot of what appear to be derived similarities that do not look like what we think the ancestral state of these skulls is. And typically, if you're a mammalogist, you appreciate the reliability of what are called basic cranial characters, at least in marsupials. These have been characters that tell us things about phylogeny that have tended to be corroborated by other sources of evidence, as opposed to some less reliable kinds of characters like the bumps on the teeth or external characters, pelage characters, and things like that, that always seem to give us the wrong answer, at least in marsupials. Well, over here is the numbat. And if you look closely at this skull, it's pretty bizarre. There's 12 teeth there on each tooth row, and they all look the same. These two guys have incisors, canines, premolars, molars that look like typical mammal teeth. These guys have little strange peg-like teeth that really can't do much. They're anteaters. They eat with their tongue, and it's not even clear what those teeth do. So just the number of teeth and the pattern is not reliable. And there's a whole bunch of other things on there too. The set of characters that we really thought we had this right have to do with the pattern of holes for nerves and blood vessels that occur around the ear region. And we thought that these characters put phyllocinids and dasyurids together as sisters. And in fact, some of the early mitochondrial data that we gathered suggested the same thing. And now we've got a completely contradictory answer that seems to be well supported. And is this just an academic point? Well, maybe not, for reasons that I'll come back to in a minute. That delightful animal is called a cultar. It's the only member of the genus Antechynomys, and tell me that's not the cutest little thing you've ever seen, except for a numbat. That little guy is about this big, and it's a marsupial jumping mouse. If you were a North American mammalogist and you saw this thing at a distance, you'd say, "Oh, look at that jumping mouse," and that's the way it behaves. And that jumping habit, these hind limbs here, with that structure that looks like a jumping hind limb, has always distinguished this animal as a member of its own genus. There is no other Antechynomys species, and it has this long tail. Well, from some of the very earliest phylogenetic results that we got from DNA, we realized that although this is a spectacular animal, it's perhaps not as phylogenetically different as it's always been assumed to be. In fact, it appears to be the closest relative of this specialized and relatively rare dunnart, the long-tailed dunnart. When the molecular results are put in front of you, you can see a lot of anatomical similarities between these things, and it's really only the jumping hind limbs that distinguish the cultar from its sister species here, the long-tailed dunnart. And then there's the third genus of Spinthopsins, these tiny little Ningawis, just a couple of grams as adults, just a couple of grams. I once nearly threw one away looking through a museum collection because I thought it was a cockroach. These things have been put in their own genus, but what we found, if you notice the mixed up green colors, was that they're stuck right in the middle of the large genus Spinthopsis. So what this is telling us is that there's some disconnect between the way we classify species in that tribe and their actual phylogenetic relationships. And there's still a lot of work to be done to figure out what those are, those phylogenetic relationships. The last piece of controversy I'd like to tell you about have to do with these animals. These are rat-like marsupials from the island of New Guinea. These are Murexias. There are 6 species of them. If you were to go back to the big tree that I showed you, they would fall out something like this, as all the descendants of a single common ancestor that are part of the Phascogalini tribe. But they are clearly each other's closest relatives. And I mean clearly. They're not even that different. They're genetically very, very similar to one another. And this has been a consistent feature of DNA phylogenies over the years. However, there is a rival hypothesis that makes us scratch our heads. This is a tree that is derived from from a bunch of those anatomical characters that I just told you are somewhat suspect in mammalogy. And it's out there in the literature, and the result of this phylogeny of these dubious characters is that the Murexias go down here at the base of the tree as if they were the earliest branches, very old and divergent branches from all the other Dasyurids. And because of this placement, the morphologist that produced this tree gave them all different genus names. They put them all in different genera, Phasca murexia, Murexichinus, and so on and so on. And in the molecular community, we sort of take a dim view of this. This is Mike Westerman's characterization of what's going on here. That this phylogeny on the right is based on so few characters and such a sloppy analysis that it really has no currency, or shouldn't have much currency in the modern literature. And it doesn't amongst phylogeneticists, except that it has made its way into the official checklists of names of mammals of the world. And that has consequences. It has big consequences for conservation because, as Robert May said some time ago, taxonomy may be destiny. When conservation priorities are set, conserving a bunch of genera is often considered to be more important than conserving a single species or 2 or 3 closely related species from a single genus. And so this is an ongoing source of controversy in the conservation of these mammals. They are not yet highly endangered, but they live in a place, a habitat, New Guinea, that is certainly not a stable place to live. So we expect that they will be sources of conservation concern soon. And we'd like the conservation planning to reflect their actual relationships. One of the things that we would like to do is be able to put dates on our tree. When, if we assume that we know who's related to who and where the ancestors are on the tree, then when did this happen? Over the course of evolutionary time, how long ago did these ancestors exist? When did the speciation events occur that produced all these clades? You're looking here at the fossil record of the order. Every dot represents a fossil genus, or in the case of the Dasyurids up here, some divergent species in extant genera. And so you can see that the record stretches back almost to the beginning of the Oligocene. And here's all those thylacinids that I told you about. The Dasyurids seem not to diversify until somewhat later. No numbats until the present. Years ago, when we attempted to use some very simple techniques to put dates on our molecular trees, the molecular clock technique, this is what we got from mitochondrial data. That's what we thought was the timing of evolution in this group. We thought that the group began to diversify back in the middle Oligocene, that thylacinids and Dasyurids were each other's closest relatives, and they split back in the late Oligocene, the latest Oligocene, that the Dasyurids didn't really begin to diversify until the middle Miocene, about 15 million years ago. And this fit nicely with what was called a faunal transition between thylacinids and Dasyurids in the same habitats. What was Australia like at these various time periods? Back at the beginning, much of Australia looked like this. A rainforest habitat, a wet, lush woodland. And that gradually gave way through the middle Miocene to something much drier, and then rather abruptly, at 15 million years ago, Australia, as the continental plate, rammed into the Southeast Asian plate, and the New Guinea highlands got lifted up in a relatively short period of time, creating a rain shadow effect across central Australia. It was the big drying. And it didn't seem like a coincidence to us that as the climates and the habitats changed from wet to dry, so did the faunal composition of the mammals. Basically, the thylacinids giving way to the more arid-adapted Dasyurids. Man, that was a good story. It was such a good story. I've told that story about 100 times, and apparently It's not right. Here is a dated phylogeny with the full data set that I just showed you, using more explicit statistical techniques that don't rely on this restrictive assumption of a strict molecular clock that, that ticks like a stopwatch. And if you look at some of the time points on this tree, the order itself seems to stretch back 40-some million years ago. The deeper branch points are still back in the Eocene. And even the family Dasyuridae itself begins to diversify way back in the Oligocene. This is not consistent with Dasyurids kicking the phyllocinids out of dry habitats. It was all wet back then. There are no Dasyurid fossils that are that old to speak of, so we don't have any way to check this. The Dasyurid subfamilies and tribes themselves really don't start diversifying until the latest Oligocene. And then the modern diversity of the group is really packed into 20 million years, which is close to what we thought, but that 15 million years ago date for the rise of the New Guinea highlands, that's pretty firm. And if the Dasyurid tribes really started diversifying before that, our beautiful story about an arid-adapted radiation goes out the window. It's not salvageable. So we're looking around for something else. And finally, I'll leave you with this. We've been studying these things all this time. And even as we've been studying them, one of the saddest things is to go back to Australia now and find that animals that seem to be common, or were common, when I first started studying them, are now facing severe threats. Of course, the thylacine is gone. The little dunnart there is the Kangaroo Island dunnart. It lives in low woodlands on Kangaroo Island, which is just off the coast of South Australia. Kangaroo Island is now a popular tourist resort, and there's very little native woodland left. Habitat loss is taking its toll. You've heard about the deadly facial tumor disease that's driving Tasmanian devils to the brink of extinction. So, in a sense, we're racing against time to try and get the story right while we can still get our hands on some tissue that's not from extinct animals. All right, thank you.